Well, I'm going to go ahead and there we go. It's just 7:30, so I'm going to go ahead and introduce Chris. Chris Cali, it's really a pleasure for me to introduce him. He's the Nicholas T. Zervis Associate Professor of Neurosurgery at Harvard Medical School. No small feat. And he's Chief of Pediatric Neurosurgery at MGH. And he came to that position in a great he's got a great history in his academic record. He went to the University of Chicago where he majored in biology and philosophy, but also was the punt and kick returner for the football team. He then went to Yale, where he not only got his MD, but his PhD. And he worked with Richard Lifton, who was like king of genetics, and then got connected with Murat Ganel and very wisely connected genetics with neurosurgery and has become one of the thought leaders nationally in this. He came to MGH for his residency, where he continued to do his passion of genetic work, and then a fellowship at Boston Children's, and was recruited by Yale, went back to work where there's a strong genetic department in collaboration with neurosurgery. But in 2021, MGH recruited him back, and that's why he's the head of pediatric neurosurgery at MGH. And it's a great example of someone who really started with an interest in an area and has married that with neurosurgery in an incredibly successful way. And he's going to talk to us today about convergent neuroscience to elucidate pediatric neurosurgical disease. And Chris and I have collaborated on a few things, had a lot of discussions, and you'll see that he's been working not only in hydrocephalus and pediatric issues, but vascular disease, tumor work, and hopefully this might spark one or two of you to look into this further. But Chris, thanks very much for doing this, and please begin. Thanks.
Thanks a lot, Dr. Ogilvie. It reminds me of when during my interview for the MGH neurosurgical residency, when Dr. Ogilvie accused me of using steroids during the interview. I weighed about 30 pounds heavier at that time.
I do not recall that, but okay.
It is absolutely true, but I have no disclosures. The funding here supports our work. Pediatric neurosurgical diseases, generally speaking, we do a lot of good work, but we do it with admittedly some pretty antiquated methods, and we still do, whether it's performing a decompressive hemicraniectomy to save someone's life from malignant cerebral edema, for putting a catheter through someone's cerebral cortex to drain fluid to treat hydrocephalus, and less so now anatomic hemispherectomy, of course, but lopping off major portions of the brain to treat medically refractory epilepsy. So we do good work, but there's room for improvement, obviously. So pediatric neurosurgical diseases, from a clinical standpoint, totally fascinating. I think it's the one part of neurosurgery still where you have to be responsible for the biggest breadth of cases: cranial, spinal, vascular, CSF disorders, developmental anomalies with challenging and small anatomy. Many of these are sporadic, congenital, structural disorders. And if you take something like Hydro or Chiari, a lot of these radiographically are going to look similar: big ventricles, cerebellar tonsillar ectopia. But just as well as I how clinically heterogeneous this population is, despite similar looking radiography. So the question is, what is accounting for those differences? Can you predict them with other biomarkers, other methods to help inform prognosis and come up with, tailored treatments? These systems, which regulate the homeostasis of cerebrospinal fluid, of vascular tone, of posterior fossa size, are extremely complex. And so the question is, if you're interested in digging into these diseases, digging into these problems, what's your entry point for figuring out where to start? Do you just pick your favorite gene of interest? Do you pick your favorite pathway that you might think is interesting? And we've taken a agnostic molecular genetic approach, starting with human patients. It's unbiased. We basically say, we don't know what's going on. We don't understand the biology. Our only bias is that in a significant subset of these patients, that there is a genetic driver. And there's a lot of reason to believe that. We exploit what we call the human model system. We start with patients because we know our findings are going to be automatically relevant for our clinical practice. So besides identifying pathogenic drivers of disease, I just think this is a fascinating field because you're illuminating fundamental aspects of both brain development, vascular development, interactions between the two. And our overall goal, I think, is to come up with a more precise diagnostic classification scheme. So I'll get into Hydro in a second, just because it's one of the more mature projects in the lab. But the idea of, coming up with a classification system of Hydro that's better than obstructive versus communicating, or normal pressure versus congenital. And again, the goal here is to come up with actionable findings that make differences for patients. So whether it's being more informed and honest when you're talking to a mom about the prognosis of their kid, or whether it's coming up with a non-surgical, SRS treatment for a brain AVM, which I'll get to later. Big paper just came out in the New England Journal 2 weeks ago for that. I mean, this stuff is really happening. And I believe in our lifetime, God willing, there's going to be some pretty cool findings for neurosurgery and for our patients. So we've set up this platform where we gather a lot of data from a lot of patients. We do it with our own practice. We do it through collaborators like Dr. Ogilvy. We do it through social media, which has been extremely powerful for us. So we're not limited in geography to Boston or New Haven. But we're gathering genomic information largely through exome sequencing, but also now through whole genome sequencing, transcriptomics, especially single cell, single nuclei, RNA-seq, and now spatial transcriptomics, which allow us to get ideas about not only what mutations are impacting these diseases, but where the genes that are mutated, where they're functioning, how highly they're expressed, and then that can give us ideas into how to set up model systems to further these hypotheses that we discover in human patients. And of course, phenomics. This is something that I think a lot of that we do a really good job at. And I think it's because we're clinicians primarily and scientists secondarily. So, a lot of the genetic studies will, identify mutations for a particular disease, but we're really interested in making radiogenomic correlations. So, okay, for patients with this sort of gene mutation, what does their MRI look like? And of course, AI and machine learning are just beautifully designed to help make correlations with this, whether it's picking up features on an MRI that you would otherwise not, you would gloss over, or scouring the medical record for coming up with other correlations that, again, we're not looking at, whether it's a type of Moyamoya disease that's associated with thrombocytopenia, as we discovered with Ed Smith over at Boston Children's, or something else. So we integrate this information to not only find mutations, but where that gene is functioning in time and space, and then breaking down and making correlations between genotype and phenotype. This is our lab website. It's a tool. It's not just to, pump out, news about what people are doing, but it's a portal where physicians, patients can contact us, enter in information in a very secure way, and get involved in studies. So if you have patients that you're interested in, you can refer them here. If you're interested in whatever cohort or disease maybe you've collected as a resident, or you're interested in doing it, come in and start getting some work done. I think our lab history is such that with the pipelines and platforms that we've set up, and it's taken time to set that up, but now that they're up and running, we've got a very good history of providing a pathway for whether, if you're a medical student trying to get into neurosurgery, you're a neurosurgery resident trying to land a job, whatever, is that you can make fundamental contributions to these projects. And you'll see that by a bunch of first authors on these big papers that are now, you know, residents at the Barrow. We've got 2 there right now. They're all over the place. So I'm really proud of that. And I think if there's going to be any legacy on my part, it's going to be trying to emphasize the contributions that neurosurgeons can make in genetics and how this is going to impact patients. So hydrocephalus, these are a couple of examples of the last couple of years in which we've used the techniques that I've described to make some, what I think is some pretty interesting findings. Nobody, you know, wants to talk about hydrocephalus at 7:30 on a Wednesday morning, probably, but I'll try to do my best to keep people from falling asleep here. You guys know the classification system. It's, I would say, a working definition that is useful for making some clinical decisions, but it falls apart pretty quickly. Basically, you know, right now, at least, you know, my practice, most people's practice, is that patient comes to the emergency room with maybe a big head, big ventricles. You're first going to want to decide whether or not this is obstructive or communicating in nature. And after that, you're going to use either endoscopic third ventriculostomy plus minus choroid plexus cauterization, depending on the patient, the etiology, et cetera, or, you know, one of the different types of shunts. So that's essentially the clinical breakdown. And if you take care of a lot of patients like we do, especially across the age spectrum, I think if there's anything going for practicing pediatric neurosurgery at Mass General, I think 2 things. One is that we do a lot of cases, and, you know, Bill Butler and I are the pediatric neurosurgeons there, and I can definitely say for any, you know, we're doing more cases than any one person, any one neurosurgeon at Boston Children's, just because there's only 2 of us, and there's still a lot of patients. And 2, for good or for bad, all the patients at Boston Children's, you know, when they're 18 years old, we have a very nice pathway where they come over. And so we're taking care of pretty complex transitional age and also adult age patients with developmental, you know, anomalies. And you know that taking care of these guys gets more difficult over time because of how many surgeries they've had. Then you layer in all the socioeconomic problems, the psychiatric. It's a complete mess, but you can make a huge difference. And so it's fun and it's exciting. So that's a bit about the clinical practice. But okay, clinical breakdown of hydrocephalus. Until I would say maybe 5-7 years ago, from a genetic standpoint, the etiology of the overwhelming majority of sporadic congenital hydrocephalus cases was just essentially unknown. You have, you know, a couple of the board exam examples of L1-CAM mutation and, you know, abducted thumbs, and, you know, some very rare syndromic forms, but the overwhelming majority of cases were unknown. So we set up this now a international consortium called HydroSeq, where our goals were pretty ambitious, but they were to collect and really characterize with deep phenomics the largest congenital hydrocephalus cohort in the world. And I can definitely say we have that now with close to 2,700 patients with some pretty robust clinical and radiographic detail. So after putting that cohort together, our goal was really to define, using exome sequencing predominantly, now whole genome sequencing, and now morphing into somatic variant detection by using little pieces of the brain that we are allowed to gather at the time of shunt placement when you make the tiny corticectomy for the ventricular catheter, to define the genetic landscape of congenital hydrocephalus. Not only that, but then to validate and then study either the most frequently mutated genes that we find, or ones that we think are of particular biologic or translational value. And I'll give a couple of examples of that. So this was, you know, back now 4 years ago, of Peter Jin's paper in my lab, where he took a relatively small cohort of 381 congenital hydrocephalus patients and found that in these sporadic cases, so cases that you would not normally think of having a genetic contribution, that close to 20%, now we know 30% with the data that I'll show in a couple next slides, are due to this a genetic mechanism called a de novo mutation, where the mutation is rare, it's large effect, it's not transmitted from mom or dad, but happens during the early course of development, and depending on what tissue that mutation occurs in, whether it's germline or other somatic tissues. For brain AVMs, for example, these somatic variants can lock in on just endothelial cells or parasites. But for here, germline de novo accounts for close to 20% of patients in that cohort. And there were a couple genes that popped up, some totally novel, others you probably recognize from cancer and some other neurodevelopmental conditions. And now when we expand this to close to 2,700 trios, this really big genetic study is in preparation. We're putting together, we call this our Hydro V3. This is our third take at it, but now with 6x number of trios, where we've really systematically defined at least the de novo contribution to congenital hydrocephalus. And I think the thing that was surprising to me is that I was expecting a different pathologic mechanism for the majority of these patients. And my previous lifetime in Rick's lab down at Yale, is when I was a grad student there, had discovered a couple of ion channel genes, and then the mutations that cause some rare disorders in fluid homeostasis in the kidney. So I was interested in fundamentally in ion transport, fluid transport, and I thought, wow, the choroid plexus, hydrocephalus, this is like the brain's version of the kidney, and it's just going to be this great theme through my scientific career of from kidney to brain, and then these mutations causing increased CSF production. But that's really not the mechanism at all. And the biology is always going to be way more interesting than you could even imagine if you allow yourself for those discoveries to be delivered to you by taking kind of an agnostic approach. And so these congenital hydrocephalus genes are not regulators of ion transport or CSF production across the choroid plexus, but instead they're fundamental regulators of neurogenesis, really multiple aspects of neural stem cell biology very early in gestational development.
Chris, could you just go back a slide?
Yeah.
Part of my ignorance. Can you just explain as you go further off the observed versus expected graph? Because what you've shown is as you get more data, that line becomes more solidified. Is that correct? Or
yeah, now that
what is that showing us there?
Yeah, so what this is really showing is, anytime, so there's a baseline rate of mutation for any gene in the genome. There's 20,000 genes in the genome. And so there's a baseline rate of a genetic variation. Some genes, based on their evolutionary significance, have been found to be relatively protected from variations, right? So you can imagine you don't want genes being mutated, especially for fundamental aspects of biology like brain development. And so what this shows, the so-called QQ plot, is just showing the relative statistical expected variation rate versus what we actually observed by doing the experiment. And so the sort of rates that we're seeing for the de novo mutation statistically would be like the chance that this is just a spurious finding would be like getting hit by lightning 3 times. And
I got it. So the further you are, the more fixed it becomes as the defect. Okay, great.
Correct.
Yeah.
So so these neural stem cell regulators, highly mutated in hydrocephalus, are expressed very early on in embryonic and then really mid-gestational development, ventricular zone, subventricular zone. There are a few cilia genes, no doubt about it. But really, when you look at those genes, I think it is, and we've demonstrated this in some model systems, it's less about cilia on the ependymal wall propagating, CSF flow versus cilia serving as transducers of signals through growth factors, et cetera, in the CSF to neural stem cells in the subventricular zone. And so this just underscores, this GO pathway analysis that looks at the genes that we term genome-wide significant hits and high confidence genes. They're highly enriched for regulators of neural stem cell development. I'll give two examples. This gene called TRIM71, it's a fascinating gene that was first discovered actually in worms. It's a stem cell regulator in C. elegans, and then in humans, it's probably one of the earliest genes and is really a marker of neuroepithelial cells. These are the very early neural stem cells that line the developing neural tube that, of course, then becomes the developing ventricular system. And these de novo mutations just don't occur kind of randomly throughout the molecule, but they're centered in these things called NHL domains. And then not only within those, not only just in NHL domains, but within those domains, particular residues that show recurrent mutation. So the likelihood of having these highly evolutionarily conserved residues to be mutated in recurrent fashion like this in unrelated patients is extraordinarily small, kind of getting back to what Dr. Ogilvie question was with the QQ plot. And of course, we've gone on to then not only do computational and statistical genetics to define, these congenital hydrocephalus risk genes, but then get at the mechanism functionally, validate them in mouse models. And so this is really satisfying when you go from a patient, one of your own patients, to discovering a mutation that's causative of their disease, and then actually able to be to recapitulate, that patient's mutation in a mouse, to have that another species develop hydrocephalus. One of the first mutations that TRIM71 mutations that we found was in a congenital hydrocephalus patient from Bridgeport, Connecticut. All right, and so then all of a sudden now we're recapitulating that mutation for that patient of mine in a mouse, and can use that as a model to understand why that patient is having disease. So whether it's engineering in one single point mutation in the NHL domain of TRIM71 causing hydrocephalus, or depleting TRIM71 in a specific subset of neural stem cells defined by EMX1 or Nestin, we use both of those Cree drivers for modeling these. You can cause hydrocephalus. So the point mutation is sufficient, and then its deletion in neural stem cells is sufficient. This is the neural stem cell work. So I think what's interesting, I'm not going to get into the mechanism in too much detail for this crowd, but I think the point is that these mutations in the NHL domain of TRIM71 screw up the RNA homeostasis of a couple different important targets that are critical for regulating the growth and proliferation of neural stem cells. And so what that does is it creates a biomechanically different cortex, because there is a severely depleted cortical mantle. There's just less neurons. There's less neurogenesis going on. And so you have a very floppy cortex, where if this patient comes down to you in the emergency room, they may, let's just say they have like a 90% head circumference for their age. Okay, maybe they're a little, they vomited a couple of times the last couple of days. Okay, and then maybe their fontanelle is full but not tense. Okay, and then they get this scan done in the emergency room. You're called down, and the question is, what are you going to do? What are you going to do? And based on how big these ventricles are, and then maybe some of these other correlative signs, a lot of people are going to do something, right, from a neurosurgical standpoint, whether it's a shunt. In this case, it's communicating hydrocephalus. But we know now through our work that these patients, this shunt really doesn't do much. It certainly doesn't decrease ventricular size. It doesn't improve neurodevelopmental outcomes. You can make someone CSF diversion dependent, that's for sure. These patients can then bounce back to the emergency room, every time, they have a viral illness, and you're going to end up tapping the shunt. You're going to, you're going to get tortured for the rest of this kid's life and the family's life. So the question is, there are these subsets of patients where ventriculomegaly, like even extreme ventriculomegaly like this, is really reflective of cortical pathology and is really a neurogenic insult. And there's multiple genes that kind of fall into this category, and it probably also accounts for the increased incidence of ventriculomegaly in some other neurodevelopmental and psychiatric conditions, right? Autism spectrum disorder, for example, there's been some great studies that have come out in super high impact journals the last couple of years, showing ventriculomegaly is actually very common in subsets of autism spectrum patients. And so that's one mechanism. Another mechanism, kind of what I call the flip side, is these recurrent mutations in the PI3 kinase pathway, whether it's PIK3CA, mTOR, PTEN. You guys know this pathway very well from cancer and some from some other stuff. Tyrone Dispenza, this PTEN paper that I'm just mentioning now, 2 days ago, just got accepted at Nature Neuroscience. He's a intern at Duke Neurosurgery right now. So this is all done by him when he was a MD PhD student, and he's a neurosurgery resident intern now. His picture is down there. But basically PTEN, highly mutated de novo mutation in congenital hydrocephalus. And it's, of course, mutated in the germline in other neurodevelopmental conditions. I've got a patient that at 9:30 this morning, we're going to be doing a combo endoscopic third ventriculostomy and then cerebellar biopsy for likely Laramide-Décluse disease. Of course, that is somatic variation in PTEN in a subset of cerebellar cells. This is different. This is in the germline. So the idea that the same gene mutated in different cell types can give rise to different phenotypes is well known, and that's what's going on here. So Tyrone modeled PTEN, same sort of paradigm that Zwi used in TRIM71, where he created mice with a single point mutation, one of the more common ones in PTEN and congenital hydro. And then based on the omic data that we have, we know that PTEN is highly expressed in this subset of cells indicated by the NKX 2.1 marker. So a neural stem cell population, NKX 2.1 positive. So again, just like Zwi, whether you change a single amino acid, right, in the genome, or deplete PTEN entirely from a specific neural stem cell population, you can recapitulate obstructive hydrocephalus. And the mechanism here is pretty cool. It's the complete opposite of TRIM71, in that TRIM71 impaired neurogenesis, decreased size of the cortical mantle. Here, there's like a cancer proliferative phenotype with increased PI3 kinase and mTOR signaling, increased neural stem cell growth and proliferation. And so you get this mass of cells around the aqueduct that just kind of squelch it off, and you develop obstructive hydrocephalus. And the cool thing about Tyrone's work, you can hopefully see the paper in a few months here, is that he's able to antagonize these phenomenon using mTOR inhibitors, right, clinically available, everolimus, for example, or rapamycin, in mice, through genetic antagonism, through mating these mice with raptor. So a good genetic pharmacogenetic demonstration that you can actually reverse an early developmental process and treat hydrocephalus with a non-surgical means. So this is a kind of a neural stem cell paradigm of congenital hydrocephalus that bears out for a significant a significant subset of patients. This de novo mechanism impacting neural stem cell regulators probably accounts for maybe a third of cases. So there's still a lot to there's still a lot to discover, but we're pretty excited about this work. So the idea that brain ventricles are not just this vacuous space, but that their enlargement can be an indicator not only of a potential fluid problem that we need to do something about as neurosurgeons, but really a signal that's saying, hey, there might be something going on, not only in this kid's fluid space, but actually in the brain parenchyma. And this has real implications for how we counsel patients and families about their expectations about when you're treating a child, with this disorder, right? And so if you can say, well, I'm going to be doing this ETV, to prevent fluid accumulation. I'm going to place this shunt, as a life-saving mechanism or maneuver. But to believe that is going to necessarily impact neurodevelopment positively, that is not necessarily true. So the fact that there's genetically encoded impairment in the brain cortex, in addition to a potential CSF problem, is really important to communicate. And I think we've gotten to a point now. This was our first take in 2020, but we did another JAMA paper in last year, really showing that exome sequencing should be a diagnostic adjunct even for a neurosurgeon. When you're seeing these patients in your office, it can be extremely helpful in thinking about how to talk to patients, how to interpret clinical presentations, especially as they get older. And this paradigm, I believe, will probably bear out for a lot of other disorders that we treat. I think Chiari-1 malformation, which we're studying in real time, is going to be a very similar paradigm, right? Where we say, okay, based on this genetic information, this patient, in addition to a suboccipital craniectomy and a C1 Lamy, needs a cervical fusion because they have some connective tissue disorder that's genetically defined. And we know that these patients, in several years after their initial Chiari decompression, are going to go on to need this. And so I think that's just another example. Another study that we did in collaboration with Chris Walsh is defining the genetic basis for a medically intractable mesial temporal sclerosis. So this is the most common indication for neurosurgical treatment of epilepsy. And so in a significant subset of patients, we currently have a patent that's being worked on right now through Harvard, which is extremely painful. But so we've been identifying activating mutations in the RAS pathway in a significant subset of patients, close to half now, where this, again, it's almost a cancer-like phenotype that this really sets us on a potential foundation for using some, what we would think of as anti-cancer agents as anti-epileptics, and transforming this from a temporal lobectomy disease to a medically managed disease. And so the mechanism here is somatic mutations. So mutations that arise spontaneously in particular cells, not in the germline, but in a localized, area, in this case, in the hippocampus. I'm not going to talk too much about arachnoid cysts, but I brought this up just to say that we're interested in everything that could possibly be related to neurodevelopment and falls within the clinical spectrum of pediatric neurosurgery. Adam Kundashora, first author paper, this Nature Medicine paper, where he defined the genetic basis for arachnoid cysts. This is fascinating because these are just, they're annoying lesions, right? You get consulted about them. Sometimes they can look absolutely obnoxious on the scan. And the obvious question I had was, what's going on there? They're always in the same, spot, middle temporal fossa. You know, there's PFAS assists and stuff like that, but the question was, wow, this is such a radical lesion. What's going why? What's going on? And so Adam took this on as when he was a Yale neurosurgery resident. He went on to become a CHOP pediatric neurosurgery fellow, and he just signed on as faculty there, where he'll be doing both pediatric scoliosis, and he's a very talented surgeon, some of the vascular stuff there. So what he found is that, again, de novo mutation highly enriched in these patients. This was a 617 patient cohort of arachnoid cysts. And what we found is that these de novo mutations really hit chromatin modifying enzymes early in development. The thing that was fascinating to me that I didn't really know about is that, you know, if you look at in the animal kingdom, you know, humans and primates are the only animals that are, you know, or species that are getting arachnoid cysts. All right, and the reason is the incidence of arachnoid cysts increases with the amount of gyrification and sulcation in the brain, right? Because the arachnoid needs to fold and develop in this very intimate way as the folding of the brain happens. And so, you know, the tough part about this is that you can't model it in rodents because they're smooth-brained, right? You'd have to use some sort of other, you know, primate mechanism. But what these chromatin modifiers are so important for are for orchestrating these gene transcriptional programs that we know are so fundamental for aspects of brain folding, gyrification, sulcation, you know, the around the sylvian fissure, the development of the sylvian fissure, that's actually why we think that most of, you know, a ton of arachnoid cysts are occurring in that area in the temporal fossa, just because that's the main fissure in the brain. And so you can find that these chromatin modifiers, again, are highly expressed in the mid-gestational cortex, and then beyond that, you can actually break down these arachnoid cysts into different clusters that have different anatomic patterns, right, based on the genes that are being mutated. And not only that, but if you really start digging into this literature, you know, this can also help explain a little bit why subsets of these patients with arachnoid cysts have other, you know, if you know, the intuition that you have as a neurosurgeon is that, man, some of these patients are kind of weird, crazy, you know, what's going on with some, you know, neuropsychiatric phenomenon. A lot of them have epilepsy, right, or seizures. And so you're caught in this conundrum as a pediatric neurosurgeon a lot, where you might have a temporal fossa arachnoid cyst in a kid with epilepsy. And then the question is, you know, do you need to fenestrate this to somehow treat the epilepsy? And I would say the answer, generally speaking, is no. And the reason is because these mutations that are causing the impaired, you know, that are driving arachnoid cysts pathogenesis are also disrupting genes in critical circuits that are, you know, related to neuronal excitability, et cetera. So fenestration or shunting or whatever of that arachnoid cyst, you can maybe make the MRI look a little bit better. But if you're talking about improving developmental outcomes, if you're talking about potentially treating epilepsy, you have to be extremely careful about that. And I think now the genetics, you know, backs up, you know, the neurosurgical intuition that, you know, unless this is causing obstructive hydrocephalus or, you know, something like that, it might be best to lay off a little bit. And then lastly, this is, you know, clearly what what Dr. Ogilvie is most interested in. I think that this is going to be, frankly, if I think about, okay, are we going to be given drugs or targeted treatments for folks with arachnoid cysts? Probably not. Congenital hydrocephalus, can we reverse that process by, you know, gene editing, you know, in you know, a fetus or something? Maybe. That's probably far off. But for vascular malformations, I think that in near term, this is something that we and neurosurgeons, we are going to be hopefully the ones to kind of push this. But We've done a lot of work with Vena-Galen malformation in collaboration with Ed and Darren Orbach, Chris, all these guys, looking at and finding that in this most severe and common neonatal high flow AVM or arterial venous communication, that mutations in the RASA1 FB4 complex are incredibly frequent. And the knowledge of this, all right, we've studied now probably about 250 patients. This is obviously a very rare condition. But we're getting to a point now and kind of experimenting with Darren. I'll talk to Dr. Ogilvie about this later, where we're able to get genetic material from the endovascular catheter, and we could think about, for example, doing while you're doing the angio, using that genetic material for diagnosing whether particular mutations, which are somatic variants, for example, in brain AVMs and KRAS, whether that particular mutation that you would not otherwise be able to know from, blood or cheek swab or something else, has a mutation that might render it susceptible or vulnerable to an agent that can, treat the AVM. So this, of course, a few years ago now, a couple of different groups identified somatic activating KRAS mutations and not only just in KRAS, but the particular G12C mutation in KRAS in an overwhelming majority of brain AVMs. Like this accounts for, probably over 60% of brain AVMs. And now this paper that I had mentioned earlier in the talk, came out I think 2 weeks ago now, where they modeled that mutation in mice, and then in 2 patients with systemic AVMs found that targeting this mutation with a drug can radically treat the AVM. And so the question is, especially for those patients that are not amenable to resection, Paul Chapman and I do really the SRS cases for pediatric AVMs, at least over at Mass General now. There's a large, at least we get a large number of patients, both from BCH and really everywhere. And is that a good subset of patients that we could think about, using this drug as either a standalone treatment or as a potential sensitizer, to the radiation treatment. So I think that that's a potential exciting avenue where genetic discovery is going to lead to translational impact. And so there's a lot of future directions, not only for the lab, but for the field. For us, continued gene discovery. I think the next big thing for our lab, we've got a 1,600 patient cohort for KIARI1. We're analyzing that now. And I think that particular phenotype really lends itself to making potential genotype, phenotype, MRI correlations. And also beyond germline analysis, looking at the role of somatic mutation, not only in vascular malformations, like I had mentioned, but also some of the neurodevelopmental conditions. So beyond that, we're going to need to model genes in a continued way for model system for testing targeted treatments, like the New England Journal paper, making genotype phenotype correlations, using AI to help us make these correlations, and not only do that, but a lot of the genetic analysis, frankly, I think is going to be done this way. And then what I'm most interested in is making insights that impact patients. And I think we've demonstrated that already with exome sequencing being something that should be kind of routine for some of our patients with newly diagnosed pediatric disease. And and then even potential medical treatments for congenital hydrocephalus. One second, guys. Delivery for our Carriage House project. Sorry, guys. So that, that's it. There a lot of work done, a lot of work to be done. Again, I think probably the biggest takeaway maybe for this crowd with the residents is that you can make, I wouldn't discount or minimize the impact you can make for these projects, whether it's Ben Reeves down at Barrow, Shrewda Fury down at Barrow, Tyrone at Duke, Emory at Michigan, Caduce at Mayo, Zwi at UVA. These are people that came through the lab, some MD, PhD students, some neurosurgery residents, some fifth-year med students, and I think that why I like working with folks going into neurosurgery, especially those who have a little bit of background in science, is that compared to some of the other students in lab, maybe the PhDs or other postdocs, you have a sense of urgency and an energy that is unmatched, and you can make a real contribution. So if anybody's interested in getting hooked up in any aspect of our work and making a bridge, especially between, Beth Israel and MGH, I think that there's a lot to do, especially with the number of vascular patients that Dr. Ogilvie has. Thanks a lot.
Well, Chris, thanks very much. Are there comments or questions that people have or want to?
Sure, we have a little bit of time. I'm Evan McNeil. I'm the PGY3 at the BI. I've got a career interest in pediatric neurosurgery and a research background in genetics, and so I'm deeply grateful for your time. I found this talk really inspiring and engaging. I want to talk about gene editing. So as you know, the best targets for gene editing are single nucleotide polymorphisms, somatic mutations, in tissues that are easily accessible by vector. I mean, we've seen massive successes in liquid tumors and genetic cures for leukemia. It strikes me that your lab has made dozens of discoveries of these single nucleotide polymorphisms, these gain-of-function mutations. And if you could just turn those off, you could create a targeted therapy for many of these progressive neurologic diseases. I appreciate the comment that you made earlier that for neurodevelopmental syndromes, the damage is often already done by the time we have access to these kids. But as neurosurgeons, we have the unique privilege to access this space, the intraventricular space, to deliver a therapy. And so I wonder where we're at right now with delivering gene editing constructs into ventricles.
Yeah, or into vascular cells, into developing cranial cells. Let me bring it up here. This is just a kind of a cute little perspective paper that we had put together. But I don't know if you guys can still see my screen, but the bottom left-hand corner, this was kind of a Eduardo.
Yes, we can.
Yeah, Eduardo Mori is he did his PhD with Chris Walsh. Chris Walsh is kind of a mentor, collaborator with me. He's like the Rick Lifton equivalent up here at Harvard that I bounce ideas around from and around with, and brilliant guy, Hughes investigator. Eduardo's a first-year resident at the Brigham, neurosurgery resident, and he did some really good work with looking at somatic variants in the context of the neurodegenerative degenerative brain in the elderly. And so I don't know if you've seen this, maybe you could read it. There's actually another good commentary put out by Ed Chang's group at UCSF that was in Nature 2 weeks ago. Something, human neuroscience, something or other. And basically, the idea is that there will be amazing gene editing success. And we have several conditions. We're very interested in taking this approach with in two different. I didn't talk about any of our work on craniosynostosis, but I think that is something that is potentially right there. I think the the G12C, G12D mutations in KRAS for brain AVMs, another great example. All right, I think that some of the recurrent mutations for congenital hydrocephalus, if we figure out the delivery issue, is an issue. I think delivery will, as you said, maybe that's something that that is your problem that you take on. But I will tell you what, the diseases that are being targeted right now are ones that people know about. But the thing that's crazy is that if you've talked to David Lude, some of these guys, the big gene editing people, about, hey, you know what, I've got a disease for which this is, I think, really going to work. And you mention it, they're like, I don't even know how to pronounce craniosynostosis. I've never heard of, this condition. And so I think that half the battle is making connections of someone maybe like you who says, okay, here's a problem in which this could really happen. And then, of course, putting in the work. The technology is there, but it's making the bridges, right? It's always going to come down to one or two people interested, passionate, who get the work done and then have the idea. But yeah, that is the next step. I think, and there's good models for brain AVM, right? Right now in mice, even some humanized models that are ready and set for it. Pfeiffer, Cruzan, craniosynostosis, it could be transformative, at least for, I think, for some of the bone vault stuff. So yeah, that's the next stage. Before you target and treat, you need to discover and diagnose. And so I think we're putting our main efforts into that aspect, at least right now. And then as time goes on, that would, God willing, be the next stage of the lab, or, maybe something that you get into.
Yeah, Chris, I think that's excellent. And also, I should mention, Chris, when he came back to Boston, we had discussions about him coming back, because at one point I almost went to New Haven, but we're desperately trying to work more together. But he has appointments at Tufts for Boston Floating, when Floating Hospital existed, at Boston Children's Hospital. He's head of genetics, neurogenetics, and at MGH. So there's great collaboration, and these projects obviously take a lot of collaboration. One question, Chris, is getting back to the diagnostic side and the sampling side, which of these diseases needs tissue, which needs cells, which needs blood? Because, there's an increasing literature in our world of endothelial biopsy, for instance.
Yeah.
They've been doing it at Yale. Some people have had success with it. Some people haven't. We're trying to figure out, is it a real thing? I'm very interested in that kind of idea. And of course, endothelial delivery of things as well. But what do we need for which entity?
Yeah, it really depends on the genetic mechanism that you're exploring. For a lot of the neurodevelopmental syndromes, congenital hydrocephalus, Chiari, different spina, myelomeningocele, spina bifida, other variants. That's a germline phenomenon that we know about most, and so that would be very amenable to a cheek swab for a blood sample. For the vascular stuff, you hit the nail on the head. This is now, for a lot of these syndromes, you can have of course, genes have two copies, two alleles. You can have one germline mutation. I'm thinking of like our HHT patients or whatever, where you have one germline mutation, and then which you can detect by blood and swab, and then a so-called second hit in one particular cell type, maybe like a brain endothelial cell, or maybe some endothelial cell in another part of your body, which of course explains why this patient with HHT has a brain AVM, this patient doesn't, but has like some systemic AVM. So Yeah, so for those patients, you're going to need localized tissue from the malformation itself, and so then that gets to the point of access to tissue. And of course, for if you're taking out one of these lesions surgically, that's an easy one. We're about to take on the KRAS discovery was really done in a few number of patients, and it does
Phenomenal, phenomenal.
does
And
represent.
this paper 2 weeks ago is just, I saw that and my heart raced. Boy, I
Yeah.
tell you, that is great stuff and very exciting.
Yeah, super exciting. So, the endothelial biopsy in our hands, we've collected a bunch of catheters from our group, and then the people at UAB. They have a lot of VGAM patients for some reason, and it's been a real challenge to get adequate amounts of DNA for sequencing. But we're definitely making progress. I think it's going to become a reality. I don't think we're going to be able to get the more or better technique from the catheter itself. I think it's just going to be innovations in DNA purification, in DNA amplification, and so we're working actually with YCGA down at Yale. I think they're even compared to the Broad, are great at this, and so that's where we're doing still a lot of our technical methodologic work. So yeah, but how cool would that be to take your patient in for an angio, figure out the mutational status of their lesion, stratify their treatment according to that, and
Well,
just
yeah, and please, I invite other people to chime in, because the other thing I'll probably talk to you about offline is Moya Moya.
100%. Yeah,
We got some work to do there, I think,
100%. So, I mean, as Dr. Ogilvie, I think, is underscoring here, he's teeing up the idea that there's a lot of room for continued work, collaboration, especially on the vascular malformation front. I'm very, very excited. We need some hands on deck for this, where we have a project that I set up when I was a pediatric neurosurgery fellow at BCH in 2015 has finally gotten to the point where we're actually able to do studies. The amount of bureaucracy and it's taken so long.
Oh, it's become increasingly difficult to collaborate, even on clinical studies, as our group knows, these data transfer agreements. And that's why you have a secure site on the web. And that's all Harvard IRB approved. And what others may not appreciate is it takes many, many months, many, many committees, and a lot of legwork to get these things in place. But when people enter that, patients and physicians, people can just, I have not been on it, I apologize. Do you get a password and then you're in, or how does it, how do you have it set up logistically? Because our residents might want to go fish around there and see what's going on.
Yeah, fish around there if you want to. You basically log in and then create a secure account. Then you email us, and then we kind of get back to you. And then by how the rules work, then once the contact is made by the patient, then we can talk on the phone. We can do consent. We can do whatever. We tell you more about the study. You can send everything is actually even populated into a REDCap database. The patient, there's a patient-centered entry, and then of course there's a parallel physician-centered entry where we get a lot of other data. We get a lot of other data as well. But let me pitch real quickly if anybody's interested. So this project really started in 2015. Was really to ambitiously say, okay, we want to sequence the Boston Children's Hospital vascular malformation experience. Basically, Ed Smith, Mike Scott for the last 30 years before Dr. Scott passed this year, I think. So we've done that. We've collected it. The tissue has been collected. The slides are cut, ready for sequencing. We have 183 brain AVMs from different patients, cut, sliced, ready for sequencing. A bunch of them we've been able to go back and recruit the folks that are, still alive, still around, to also get blood or cheek swab, for somatic variant detection. So we'll be able to systematically define, not only how frequent KRAS mutations are in brain, but what cell types they're occurring in, and then also look at, well, what's responsible for the remaining percentage of cases that have not yet been defined. Maybe those are going to be targets of a different type of drug, or maybe that might explain why, wow, we just gave proton beam, for, this type of ABM, and it doesn't do a damn thing. Whereas this other one has a radical, a radical response. Like, what's accounting for that phenomenon? So both with AVMs and then cavernous malformations, sporadic cavernous malformations, not
familial.
not CCM 1, 2, 3. So anyway, so that's just
Well,
another.
that
thing.
is, now listen, full professors may apply as lab techs, right? I mean,
Can you?
I can go over and work for Ed, right? But it's fascinating, and that is just unbelievable data could come from that. Unfortunately, our time is up, but Chris has his website, and if anyone wants his email, we have access to that. Thank you very much for doing this. It was really great, and what you're doing is just really strong work. And I as you know, I strongly encourage and support it, and look forward to future collaborations. But thank you very much for taking the time this morning. It was a great talk. Thanks very much.
Always good to see you, Dr. Ogilvie, and see you later, guys. Thank you for the opportunity.
Take care.
Bye-bye.